The first part of the cell cycle is interphase. There are three parts to interphase: G1, synthesis, and G2. In G1, or gap 1, the cell continues its normal functions and goes through a checkpoint, where it is assessed for criteria such as nutrition and size. In synthesis, the DNA stored in the nucleus replicates and gets loosely organized. This unorganized group of DNA is called chromatin. Next, in G2/gap 2, the cell essentially does what it did in G1. It has another checkpoint and continues to grow, getting ready for mitosis. In mitosis, the part of the cycle in which the cell divides, there are four stages: prophase, metaphase, anaphase, and telophase, or PMAT. In prophase, the chromatin organizes into chromosomes, made of two sister chromatids. Now before I go any further, let me break down the parts of a chromosome. A chromosome is made up of two chromatids, which have telomeres at each end. These two chromatids are connected by a centromere, and on both sides of the centromere are the kinetochores, the part of the chromosome the spindle fiber is attached to. Now back to your regularly scheduled programs. Also in prophase, spindle fibers coming from the centrosomes start to spread to the center of the cell and the nuclear membrane breaks down. Next is metaphase. In metaphase, the spindle fibers attach to kinetochores, and using polar fibers to pull on each other, they align the chromosomes into the middle of the cell. In anaphase, once the chromosomes are aligned, the spindle fibers pull apart the chromosomes into chromatids. Then these chromatids move to the other side of the cell. The nuclear membrane then reforms, chromosomes uncoil, and spindle fibers fall away in telophase. In cytokinesis, the cytoplasm separates, forming two cells. In an animal cell, a cleavage furrow forms, pinching the cytoplasm apart. In a plant, because of the cell wall, a cell plate forms, creating new resources for the plasmid membrane, eventually creating a new cell wall that separates the two cells. If all goes according to plan, the end result is two identical cells. Thus, the cell cycle starts all over again.